Hello everyone, my name is Ian Diaz-Nieves. I am a student at the University of Puerto Rico, Rio Piedras campus, and I will be presenting my research proposal developed in concert with the PI of my lab, Dr. Alfredo Gessi, called Ascertaining the Epigenetic Mechanisms of Alcohol Dependence, utilizing a CRISPR dCas9-based toolset. Based on the homeostatic theory of drug dependence, we can consider our brain to behave like a scale. Exposure to an addictive substance places a weight on the scale, causing a one-sided imbalance which we call the drug state, during which the effects of a drug are fully active in an organism. Repeated visits to the drug state over a period of time forces our brain to try to counterbalance the weight of the drug on our system by developing specific adaptations to directly oppose the effects of the drug. In the case of alcohol, its primary function as a depressant would be to slow down synaptic firing, so the brain responds by increasing ion channel concentrations at the synaptic terminal. However, what happens when the drug is no longer in our system? Without the weight of the drug, these changes now produce an opposite effect of equal magnitude to the drug state. This is known as the withdrawal state. Drug withdrawal can be a very uncomfortable and grueling experience, and unable to cope, most people end up relapsing into drug use to manage their symptoms of withdrawal. The changes in the brain caused by addictive drugs, such as alcohol, are long-lasting and have been shown to affect expression of certain genes for an extended period of time, even after just one exposure. This led scientists to propose that alcohol acts on neuronal cells by epigenetically changing the rate at which certain genes are transcribed through modifications of their chromatin structure. The problem? Although in the field of alcohol addiction research it is widely accepted that alcohol dependence comes about through certain epigenetic processes, such as the acetylation or deacetylation of a particular histone tail, the studies that have described this relationship have done so mostly based on a correlation between the presence of these marks and changes in mRNA levels of expression of different genes following drug exposure. This, however, does not establish causality between distinct chromatin marks at specific gene loci and the observed behaviors that arise from alcohol addiction. Clearly defining this relationship will be imperative for bettering our understanding of the mechanisms of alcohol dependence down to the level of single molecules, as well as the development of future, more effective treatments for people who suffer from severe drinking problems or from alcohol abuse disorder. We hypothesize that histone residue modifications near the regulatory regions of alcohol-responsive genes are responsible for addiction formation and the subsequent display of withdrawal symptoms. Our approach for testing this hypothesis will be by way of a CRISPR dCas9-based toolset. Firstly, we would design a fusion protein between a deactivated Cas9, meaning a Cas protein that has lost its catalytic function as a nuclease, and any of an assortment of epigenetic effector proteins, such as a histone acetyltransferase or a histone demethylase. And just like a regular Cas protein, this dCas fusion would be able to use a small guide RNA that we give it to locate essentially any gene in the genome. And once there, the fused effector's catalytic function can be employed to intentionally place chromatin marks on the regulatory regions of genes of interest, and in that way control the expression of just that one gene. This is depicted in the image to the right, where we can see a dCas9 protein bonded to the DNA with complementary sequence to that of the guide RNA, as well as the different histone residue modifications that can be made with the effector fusion. And quickly on the left, just to show another possibility of this toolset, we see how a transcriptional activator or repressor could be bonded as well to influence expression of the gene. Afterwards, the plan is to introduce this system into our model organism, the humble fruit fly Drosophila melanogaster, a staple of alcohol and genomics research. In conjunction with the CRISPR system, we also plan to incorporate the standard UAS-Gal4 system to introduce a way of activating our fusion protein with temporal specificity by using a temperature-sensitive enhancer. To test the resulting effect that placing chromatin marks around the regulatory sequence of specific genes has on the flies, we will be conducting a recovery from sedation assay in order to measure the development of tolerance to alcohol. The experiment consists of placing the flies in vials and then exposing them to alcohol vapors until all flies are passed out. Afterwards, we would exchange the ethanol vapors for just humidified air and wait for the flies to get back up while taking note of how long it takes them to recover. We will do this twice across 2 days. However, after the first exposure to alcohol, the flies will also be heat shocked to activate transcription of our dCas9 effector fusion and enable the placing or removing of the chromatin marks that we wish to study. Then finally, after a second exposure the next day, we will compare the tolerance of both sessions to determine if the flies did in fact grow tolerant to the alcohol. The setup of this experiment can be observed on the right of the slide. Although it looks simple and straightforward, performing this kind of assay can be quite labor-intensive for the researcher. Regardless of whether or not they possess a recording of the experiment, they would still have to manually go through each video file and take note of each individual fly's behavior during a session that could last hours, depending on the ethanol concentration. and creating a software capable of doing the counting for us has proved difficult because of the vials where the experiment takes place. Not only can flies cover up other flies on the opposite side of the tube, but flies can also be hidden from view in the sides of the vials due to how it curves. Because of this, another one of our goals is to optimize the classic recovery from sedation assay by developing a setup that can be easily automated with the incorporation of a very simple computer vision algorithm. This design deals with the two major flaws that the classic version has, which are fly stacking and the curving of the vials. Here, all of the flies are laid out flat inside of each chamber, making it possible to keep an eye on each and every one of them, as well as it does not allow the flies to climb on each other, virtually eliminating the possibility of a fly being obscured while in the assay. I won't be going into further detail, but please be sure to reach out if you want to know more about this kind of assay. Lastly, here's an outline of the possible results that we expect to have. We expect to find a difference in tolerance levels between our epigenetically marked flies and our control group. With our findings, we would be able to determine or not causality between the chromatin marks that were placed in the development of alcohol dependence/addiction. We also expect to finish the construction of our new tolerance assay in order to perform all of our tests on it and be able to validate its efficacy. And finally, if we were able to establish causality, then we believe it will be possible to take an individual from a dependent state back to an alcohol-naive state by removing the specific epigenetic changes made by alcohol. Quickly, I would like to acknowledge the Gezi Lab and all of its members, and I would also like to thank the NIH BP Indoor NeuroID program for giving me the tools and resources to pursue neuroscience. This has been my research proposal. I hope you enjoyed it, and this is the end of my presentation.